The adaptive immune system is a specific defense system that eliminates almost any pathogen or abnormal cell in the body. The activities of the adaptive immune system include amplifying inflammatory response and activating complement. The shortcoming to the system is that it must be primed by an initial exposure to a specific foreign substance, and this priming does take some time. The characteristics of adaptive immunity include the idea of being specific, and here your system recognizes and targets very specific antigens. It is systemic, which means it's not restricted to an initial site, and it has memory, which means it mounts an even stronger attack against known antigens when you've been exposed to the same antigen a second time. Two main branches of adaptive system include humoral and cellular immunity. The humoral immunity involves antibodies, so it's sometimes referred to as antibody-mediated, and cellular is also called cell-mediated immunity. The humoral immunity, antibodies are going to be produced by lymphocytes, and they circulate freely in the body fluids, or the humors. And they bind temporarily to the target cell, and what this does is it temporarily inactivates the cell, and then it marks it for destruction by a phagocyte or by complement. This humoral immunity is or has to deal with extracellular targets, so targets would be outside of your cells. For cellular immunity, the lymphocytes act against target cells, and they do so by directly killing the infected cell or indirectly by releasing chemicals that enhance inflammatory response or by activating other lymphocytes or macrophages. So cellular immunity is or has targets that are cellular or that are cells. So we're going to look at antigens, and antigens generally are substances that can mobilize adaptive defenses and provoke an immune response. These antigens are the targets of all adaptive immune responses, and most are large of these antigens. And so they are complex molecules, and they're not normally found in the body, so they're considered non-self. The characteristics of antigens. Antigens can be complete antigens or incomplete antigens, which are referred to as haptens. They do contain antigenic determinants, which are parts of these large molecules. And antigens can also be self-antigens. Antigens can be complete or incomplete. So looking at complete antigens, right here, they have two important functional properties. One is immunogenicity, and the other one is reactivity. Immunogenicity is the ability to stimulate the proliferation of specific lymphocytes. So immunogenicity would get your body to produce more lymphocytes. And reactivity means that the complete antigen has the ability to react with activated lymphocytes and antibodies, which are both a consequence of immunogenic reactions. Examples include foreign proteins, polysaccharides, lipids, and nucleic acids, which are seen on many foreign invaders such as pollen and microorganisms. So these large structures like pollen microbes get in you would have these types of polysaccharides, lipids, or nucleic acids on them. Incomplete antigens, which are also called haptens, are molecules sometimes that are too small to be seen and are not immunogenic themselves. And examples include peptides, nucleotides, and hormones. So while they may themselves not be immunogenic, they can become immunogenic if the hapten attaches to your body's own proteins. So the combination of the protein and the hapten is then seen as foreign. And this can create problems. So this can cause the immune system to mount an attack that is harmful to the person because it attacks self-proteins as well as the hapten. And examples would be poison ivy, animal dander, detergents, and cosmetics that can have this effect. Antigenic determinants. These are going to be parts of an antigen that the antibodies or lymphocyte receptors bind to. And the model on the right here, you have your antigen right here, and they show 3 parts to it right here, and those would be the determinants. So those would be the parts of the antigen that antibodies have been created specifically for. Okay, so those would be considered antigenic determinants. So in the key down here, this would be the determinant A in our model above. This would be determinant B based on its shape, and then C right there. Okay, so most naturally occurring antigens have numerous antigenic determinants, and these determinants can mobilize several different populations of lymphocytes because there are several determinants on the antigen, and they can form different kinds of antibodies against them. So there would be a specific antibody for determinant A, a specific antibody for the antigenic determinant B, and so on. Also, as a related nature here, large chemically simple molecules like plastics would have little or no immunogenicity or influence on your immune system.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
And then we have self-antigens. These are also called MHC proteins. And these self-antigens, these are what we see on all of our cells. So all of your cells are going to be covered with a variety of proteins located on the surface that are not antigenic to self, but may be antigenic to others in cases of things like transfusions, like a blood transfusion, grafts, or even organ transplants. So there's one set of important self-proteins, and they are a group of glycoproteins called MHC proteins. And MHC proteins are coded for by genes these genes produce proteins called major histocompatibility complex, which is what MHC stands for. And these are unique to each individual, so your MHC is unique to you. They do contain a groove that will hold a piece of self-antigen or foreign antigen. And T lymphocytes, for example, can recognize only antigens that are present on MHC proteins. So a T lymphocyte is one of the white cells that can go and recognize antigens, but will only do so when they are presented on those proteins. So what I did was I went and downloaded an image just to kind of have a visual of what one is. So here you see on the left and the right, you see 2 cells, cell membranes, and then there's your MHC protein there, and there's the groove in there that would be holding or presenting the antigen. On the right here you see 2 different MHC proteins there, each with their grooves, and you can see that they have a specific groove that they are attaching to this specific antigens there. So those would be an illustration of your major histocompatibility complex proteins.